As we continue with the Neurology Session 9, we welcome Dr. Marios Pantentzais and Dr. Nicolas Grigoriadis to the stage as chairs.
Well, it seems that I will be alone. I don't know where Nicolas is. Anyway, I have this. Okay, so we proceed to the next session and the next speaker, which is Dr. Richard Frakowiak, and his a distinguished doctor that he's dealing with cognitive functions and human project at UCL, and he's going to give us a talk about what the future is promising to us about the clinical neuroscience and the research on that topic. Thank you very much.
Thank you very much, and thank you for the invitation. I won't name everyone. It's very dangerous to prognosticate, as we have recognized last Tuesday. But on the other hand, it's also very important to prognosticate, because otherwise you're stuck in one way of thinking. So that the process is a virtuous circle which involves destruction of hypotheses. I'm going to talk today about the future. So do please attack me as much as you can. And I'm going to talk about clinical neuroscience in particular. Now, I need to know how to advance. Good. This was the situation in 2010, and I can tell you it's much more complicated now, of the amount of neuroscience data we have. Resolution along the y-axis, types of studies and types of models along the x-axis, each investigated to a high degree of precision and very few of them interacting with each other. And this is our problem. So the question is, can we help? Well, we can get to the human. There are always questions about rat models and mouse models and how effective and efficient they are. In the human, we have had non-invasive human brain modeling, imaging, and that has been very helpful. But it is simply at the mesoscopic level. But there is a nice thing happening over the last 5 years. It is becoming quantitative, something that used to happen with PET scanning, but it involved radiation. It could be repeated in anatomical terms as densities, but now we can actually measure things like water content, myelin content, regionally within the human brain. So one way is to improve our technology, and this is essentially the American way, getting more and more specific, better and better resolution, and so on. But our task is to understand the brain in its totality, the multi-scale brain, going from the molecule, a little piece of DNA, all the way up to cognition, and trying to bring together all the information we already have, and knowing which gaps we have and where we need to target our future research. So, this has been attempted by the laboratory of my friend and close colleague, Henry Markram at the EPFL, where he has understood that many people in engineering don't generate hypotheses, collect data, and reach conclusions, but they collect a lot of data, work out how, at one level of resolution, the different components work, so get the rules of the game that we've just been talking about, and then move up the system, which is now being constrained in its degrees of freedom, because at each level, if you know the rules, you are limited in how the next level can work. So I'll illustrate that with a slide that he's lent me. As you know, we've been looking for mathematical expressions of rules in neurology since 1948 with the Nobel Prize of the people who looked at the transmission of signals rapidly across the axon. These were the Huxley-Hodgkin equations. There are a lot of such models already developed for ion channels. There are many such models developed for synapses. In addition, we have masses of information about how different neurons are distributed in the cortical columns. We have a lot of information dating from work done mainly in Russia and in France in the 30s, 40s, still in the literature, about which neurons connect to which neurons at which level, and now with the amazing work going on in Seattle, we're getting really precise information about where different neurons are and how they connect. So the question then is, can we predict how the next level will work? And this has been done for pairs of different types of neurons. So on the blue side of this, you see what is found, and on the red side, you see the modeling result, so that you're trying to compare the model's function, in this case an anatomical function, with what you find in the validating anatomy. This can be done also for functionality by introducing a current into a model. You can show in vitro on the left side, in silico on the right side of the lower panel, and you can show how nicely they compare each other, either as multiple trains of impulses or single impulses, and even the whole disynaptic inhibition system. So this is the theory behind how to go about things. The development of models at different levels of organization that are driven by data and the technology of simulation, a technology which has been used by engineers for at least 40, 50 years, and which are involved in building cars and airplanes and everything. These models are mathematically expressed, and these models then have to be biologically validated. So, this is the sort of result. This has just been published in Cell Biology. It is a simulated mouse cortex with all the neurons involved in a rat. And that's all I can tell you about it, because the paper's just come out in Current Biology. But the essence of the technology is that you could now interrupt mathematically this model and see what the effects were on the rest of it. So let's get back to the human. The human at the moment doing neurology is about 140 years old. He taps reflexes, he looks into eyes, he scratches the sole of the foot. And he provides a pattern of abnormalities, and he causes a disease. Of course, this isn't a disease, as she would have showed him in the first place. It's a syndrome. So the syndrome has now reached its limits. We've just heard how different phenomenologies come from the same genetic abnormalities, or same phenomenology may be caused by multiple genetic abnormalities. So the Human Genome Project, which has been so influential, as has been described by Professor Antonarakis, is a constituent element of organic matter, but it does not provide us with a lock and key answer to understanding human disease. Modern science, I've shown you, is fragmentary and atheoretic, and can run down blind alleys like the amyloid situation we discussed previously. Modern clinical neuroscience, as again I showed you, is becoming increasingly sophisticated, and modern information technology and modern mathematics have their own problems, not least of which is the physical and energetic constraints of doing modern information technology. As you know, people like Amazon and others are building their own or buying nuclear power stations in order to generate the power that is needed at the computational level in order to generate these sorts of models. But in the end, what we must be hunting for is mechanical biological diagnoses, as has just been stated as well, so that we can then know what we have to fix. Now, the question in neurology is, do data exist that permit such mechanistic diagnoses already? And the answer I'm going to suggest is yes, and that these data are distributed in all the hospitals, clinical laboratories, fundamental laboratories in the neurosciences in the world. The question is, how can we use them? And Google has shown us how we can use them. This is not to put them in large warehouses and to put them in a specific way and to curate them and to make them all the same, but to solve that problem by using all the data, because then the signals will come out from the noise, and that is done by adressage, addressing the sum of all the data. So this is the radical concept here, which is that you need to be able to address data with your question, not you need to be able to create a warehouse which is highly stratified in order to then think about what questions you're going to ask. So the other interesting thing about addressing all the data is that there are now algorithms available that can mine the data. And by mining, we mean look for all the possible correlations and, more importantly, the interactions between those data with a statistical assessment of how well the ensuing models explain those data. So this raises the issue of whether the data can actually function also as hypothesis generators. Whereas the first is a question of having a hypothesis and addressing the data in order to see how to answer your question. And the question, I believe, that is going to be here for the younger people amongst you is to say to yourselves, we are fed up with this Bible of syndromes, which is mainly used for getting payment for your activity, which is called DSM-4, into a true Bible of disease signatures. What do I mean by a disease signature? A disease signature is a constellation of biological, physical, physiological, electrophysiological imaging data in humans, which are common to certain sets of humans, so that we can then use those commonalities to go back to the doctor and say, are these people expressing the same diseases as syndromes, or is there a wide range of their expressions, which is what I suspect will be necessary. And that will be done in the very near future with artificial intelligence, and specifically with artificial general intelligence, because we will be able to address with much greater precision and variability. Now, we already know that informatics integrate a lot of data. This is a half day's work by a postdoc 10 years ago. What's that? Oh, yeah. Sitting on a desk, addressing the National Library of Science in Bethesda, and interested in what he wanted to image in Parkinson's disease, because he knew that the subthalamic nucleus had one particular target, and that connection was the one that gave him Parkinson's disease. But he also knew that Parkinson's disease was rather heterogeneous syndrome. And so he just asked of all the literature that was there to bring out and give a picture of the connections that those two, and as you can see, the connections are multiform. There are hundreds of them, both afferent and efferent to different parts of cortex as well as to the basal ganglia, from the subthalamic nucleus, and also to the basal ganglia, back to the subthalamic nucleus and around. So this is the complexity we have to deal with, and we cannot do this purely by imagination. It's too complex. The other thing we could do is to classify patients already with informatics, simple informatics. So this is the first example of artificial intelligence, which I published in 2007, on trying to discover how can one find Alzheimer's disease before death. Now, if you make a diagnosis of Alzheimer's disease and take people to post-mortem, the neuropathologist shows you are wrong in 30% of cases, even in the very best centers. So we have to understand that there are people with dementia who don't have Alzheimer's disease. We've already discussed that. So at the top here, you have the best possible article that I could find, with a correct diagnosis of Alzheimer's of 81%, 61% sensitivity, 93% specificity. We trained a support vector machine, which is a type of artificial intelligence, with images from friends of mine in the world who had patients who had had MRI scans with a diagnosis of Alzheimer's or with a diagnosis of complete normality, who died with those diagnoses and who would send me their scans. After the neuropathologists had said, yes, this is real Alzheimer's disease, yes, this is real normality. This is not just normality which is pre-Alzheimer's, it's normal normality. And of course we found 4 groups: Alzheimer's that had Alzheimer's pathology, Alzheimer's that wasn't Alzheimer's, normal that was truly normal, and normal that had the changes of Alzheimer's preclinically. And the results from these binary classifications are shown at the bottom, 95% in all cases. Now, the line I show here is a great simplification. It's 2 dimensions. We're dealing here with multiple dimensions, tens of dimensions, who knows how many dimensions, but so that these mathematical boundaries are complex and cannot be visualized. So we need informatics. So now you'll say to me, well, how much data do we have? Well, we have all the hospital databases in the world. We know we can do this because Netflix does this every night. Moves data, there it goes there, sends it here, there, everywhere. Very quickly, you just press a button and it's there. These hospital databases are not complete, not structured, not standardized, not clean. That's fine. We're dealing with vast amounts of data if we do this. We can protect for privacy. It's not protected against corruption, so we have to protect against corruption. But hospitals have engineers who do this. Every hospital has a data security set up. We also have research databases. That's our databases. We protect these culturally because we think our database is going to win the Nobel Prize. A database has never won the Nobel Prize. It's your intelligence in questions. So we need to get with it, stop all this shutting off the databases. Open them up, put them on the web, and let's get on with it. And you use other people's data, they use your data. This is our duty. Get real. And then the final ones are the pharmaceutical databases. Well, people say he's mad. So I went to see the CEO of Sanofi. And I said, I want your databases. And he said, you're mad. I said, no. I said, what percentage of your double-blind clinical trials are positive? Oh, about 3 to 5%. I said, can I have all the rest? Oh, I see what you mean. So even they are. And they have really very well-structured databases. So at one stage, I had 5 big hospitals throughout Europe, all linked up and all doing this sort of thing. And then I retired and there was no funding, and this is all to be done by people who are a lot more savvy about informatics than I am. And the hospital management were the main supporters, because they already had security procedures. Every day in real time, they produced archives off-site to protect their data. And all we wanted was to have our own off-site archive server where we could pre-process all the data, the neuroscience data, by denoising, standardizing, anatomically normalizing, numerically normalizing, and creating a database which could be addressed by the investigator outside the hospital. And if he addressed all the hospitals in the world with the question, do women of the age of 30 compared to the women of the age of 40 at sea level or at 1,000 meters have more or less multiple sclerosis in the spring and the summer? That would be an address. He would send that, and locally they would find the data which was needed for you. Locally they would aggregate it so it becomes not break-intable, and then they would send it out to a unified portal, which we had already built as well, which would then aggregate it a second time, guaranteeing secrecy for the individual, and generate an answer, and an answer which was for the population as a whole, which a doctor could then, when seeing a patient, say, this is what you've got. Do you mind if I send it to find out whether it corresponds with any result that is online? So the doctor remains in power, the hospital remains safe, the hospital management can ask questions about which diseases when in the year, so that they can organize their buying of equipment and opening beds and all this sort of thing. And this gives you a whole series of results which can then be removed, whereas the prevalence can be stored, and so that you can get back to what you want. Now, let me just give you one example to show you that this works. 6,000 normals and Alzheimer's, completely random data from 3 French cohorts, with all sorts of data, including proteins in the CSF, genetics, not in everyone by any means, because you can interpolate if you have enough data. And we went through 3 types of analysis of this: a phenotype-led, semi-supervised clustering, a cluster analysis. This is data mining. We've heard about the dangers of phenotypes. A biologically led classification, where we know how we are working still hard at understanding the biology, or deep learning. You remember this is the DeepMind technology which won the Nobel Prize this year with Demis Hassabis. And this is one result, right? We started off with all the people in the cohort who appeared to be normal. And we found that a proportion had the pathology of Alzheimer's disease in blue, and the rest had another pathology, or were completely normal. And then we followed them in time. And those who had the pathology of real Alzheimer's disease had a completely different prognosis to those that don't have the pathology of Alzheimer's disease, but who can be demented for other reasons. And this difference is bigger than the difference between ApoE4 homozygotes. So, data mining to provide disease signatures. Just watch that big pink circle. That's all the patients with dementia, and they're associated with a beta, they're associated with a pattern of imaging. These are the various types of data that we've been looking at. There are normal people who have different, they're in blue, who are at different ages, and who also have dementias of other types. This type of thing is what we're looking for with data mining. So, I've finished. Thank you very much.
Thank you. So thank you very much for this interesting and very complicated data that you have presented.
Well, you won't do it. It's the younger people who understand.
Yeah, okay.
You have to be a non-professor to understand this.
Is there any question from the audience?
Come on, younger people, where's your questions? Yes,
Yes,
no,
Professor.
you're not young.
Okay.
Is there a younger person here with a question? Oh, this is the European Medical School. Is it high quality? Come on. What about him? He's looking away because he doesn't want to ask his question. Yeah, come on then. He needs a microphone.
Okay, at the start you said that we're still at the beginning of all of this. Where do you see it going in the next, let's say, 20 years?
No, I'm not hearing you.
At the start, you said we're still in the beginning of this, of the procedures. Where do you see it going in the next, let's say, 20, 30 years?
Well, I think, so that was my question. Well, say it again, because I'd still not quite heard it.
How do you feel time will involve to have all this way of thinking applicable in the clinical setup?
Oh, well, this was going to be my last, oh, it's gone. It was going to be my last slide. You have to stop doing things the way your seniors do it. Now, forget syndromes, except for exams, and because it will help you give current treatments. Instruct yourself or find collaborators who are real experts at informatics and experts who are now appearing who are real experts at artificial intelligence techniques, which are they're just sophisticated statistics in multi-dimensions. And then finally, find something that you want to know the answer to. And get yourself involved with a university which has, or a country which has computing facilities, but that will soon not be important. We started off wanting supercomputers in Europe, and we got 3 or 4 of them lined up. But 4 years into the project, we realized for much cheaper, we could just hire Amazon time. So things are moving very fast. I think from the point of view of the bosses, what you need to get them to do is to start introducing this culture of open science so that you can talk to people around the world who are thinking about the same things as you, and so that you could create networks to begin to do them. And the other thing the bosses need to do is to start talking amongst each other about how to sort out this problem of creating a health Google for the world. Now, Europe had that chance. It didn't see the vision, and it gave up on it. It needs people with a bit of seniority and a lot of oomph to put together another program to go to Europe and to say, look, we really need to do this. Because not only if we know it works, but not only, you've got to scale it, so we don't know if it scales. We know it scales to 5. Does it scale to the world? Well, I gave you some reasons why I think it does. I mean, you just need to think of telephony. Even telephony tells you that can be done. So you really need to go back to them and say, hey guys, you need to get wise.
Well, thank you very much.
Pleasure.
And thank you.
We would now like to invite Professor Ioannis Petrikis for him to introduce our distinguished guests.
Good afternoon, everybody. I'm going to introduce our honorary keynote speaker, Thomas Sudhoff. Thomas Christian Sudhoff obtained his MD and doctorate degree from the University of Cologne in 1982. He performed his doctoral thesis work at the Max Planck Institute, and he began his independent career as an assistant professor at UT Southwestern in 1986. Sudhoff's work increasingly turned to the analysis of synapse formation and specification, processes that mediate the initial assembly of synapses, regulate their maintenance and elimination, and determine their properties. Sudhoff served on the faculty of UT Southwestern in Dallas until 2008, and then became the Avram Goldstein Professor in the School of Medicine at Stanford University. In 2013, he was awarded the Nobel Prize in Physiology of Medicine, together with the two US-American biochemists, James Rothman and Randy Shegman, for the discovery of the vesicle trafficking. The lecture title is two words, Understanding Synapse Formation. Professor, the stage is yours.
Okay. Thank you. Good afternoon, everybody. Thank you for this kind introduction. I've never been on a stage like this. Thankfully, I took on my red shoes today, otherwise I'd be completely misplaced here. So it's a wonderful occasion to be here. This is the wrong, okay. Let's start at the beginning. So I changed my title a bit because I wanted to tell you, in a better fit with the theme of this meeting, about our attempts to try to connect molecules to an overall behavior, an organismic function, memory. What I'm going to tell you about today is going to be rather detailed, because that's how we do things. I'm going to tell you about some real experiments that we are performing, whose analysis or interpretation is not always totally clear. In my presentation today, I may lose you, but don't despair. I'll try to catch up with you, or give you an opportunity to catch up later on, because some of the data that I'll present may be foreign to you. The concepts that I'm going to talk about are concepts of modern neuroscience, where we are at this point in trying to understand the brain, using a variety of approaches, ranging from molecular analysis to behavioral studies. In neuroscience, there's a general discussion always on what is the best approach. You've just heard, for example, in biomedical research, that there's an increasing emphasis on data mining, which is very useful to some extent. Other people believe that we need to study the brain as a black box, because it's so impossibly complex that we'll never understand it. Anyway, a bit of a pessimistic view.
Others, on the other hand, are more interested in single molecules, channels, or specific neurons somewhere in the brain. Despite all these different views, perspectives, I think everybody in neuroscience will agree that there's a unifying concept, very vague, in the idea of the neural circuit. And so neural circuits, as illustrated here very schematically, are in many ways the starting point that people use to actually discuss how the brain might work. Neural circuits are formed by synapses that connect neurons into networks. The idea of neural circuits in some way ignores the fact that there's other types of cells in the brain that are just as important. Without them, the brain doesn't work either. Synapses transfer the information from one neuron to the next in the brain, but they also change that information. They're the fundamental computational unit. They change this information by transforming a given spike code in a presynaptic neuron in a differential postsynaptic response. They have amazing properties that we and others have been studying for decades. The major goal of our lab at this point is to try to understand how neural circuits are constructed, how they are formed by the interaction of neurons with each other. And when you look at a synapse, it is fundamentally an intercellular junction between a pre- and a postsynaptic neuron. As an intercellular junction, it is presumably formed by the interactions between pre- and postsynaptic surface molecules that are referred to as cell adhesion molecules, even though the major function of these molecules is not to adhere, but to signal. Many synaptic adhesion molecules are described, some of which are shown schematically on this slide here. You don't need to read all this. It's just a colorful slide of different molecules. On the left side, it shows the presynaptic side. On the right side, it shows the postsynaptic side. You can immediately see that there's many more on this right side than on the left side, because on the left side and the presynaptic side, there are these hub molecules, such as the neoexons, that interact with a lot of postsynaptic molecules. What is apparent, however, from this description of all these transsynaptic interacting adhesion molecules, that these molecules themselves form networks. There's molecular networks, just like there are neural networks. And these molecular networks presumably guide synapse formation. How synapses are formed and specified, and which molecules are actually important, is unclear. And you can't use data mining to find this out, because literature is all over. It doesn't tell you. So we and others have embarked on programs to systematically study this, to figure out how this works. And we've also tried to understand how these processes might control synapse dynamics in disease and in learning and memory. My talk today will be about learning and memory. It will be about trying to understand how synapses operate in learning and memory, which we don't actually know. You will read in textbook that synapses undergo long-term potentiation, and that explains memory, but that's, I would say, at best a simplified version of what we know, at worst misleading. We are guided in trying to understand memory by a concept whereby memory acquisition, consolidation, storage, and retrieval are separate phases that likely involve many specific circuits and molecules, and that one can actually analyze this to some extent. We also hypothesize that although LTP is an explanation of memory, maybe too simplistic, overall synapses must be essential for understanding how memory might occur. Conceptually, we divide memory formation into a number of phases sensory inputs, for example, by learning, by seeing, by smelling, whatever, leads to memory acquisition. This memory is then either kept as a short-term memory or consolidated into a storage space to become long-term memory that can be recalled and reconsolidated during recall. This is a hypothetical and unproven diagram. Memory consolidation and storage have not been experimentally separated. And where and how memory is stored remains enigmatic, as does the nature of consolidation and reconsolidation, and many other questions surrounding this. Because memory likely involves synapses, transsynaptic adhesion complexes must be central to memory formation, and I already talked about this. And I just want to point out one particular complex here that is identified on the top that I'll come back to, because it turns out that we have evidence, not yet conclusive, that this complex is particularly important for one particular type of memory. So today's talk on memory circuits will suggest key roles for this complex, as I will describe to you in following. And this is shown here again in more detail, more later. Okay, so this is what I'm going to tell you. I'm first going to tell you about what type of memory we are studying, because this is a bit unusual. 99% of people who study memory study one particular memory, fear conditioning. We are the 1% or less than 1% that also study something else in addition to fear conditioning. And I will tell you two stories that illustrate two different first facets of memory, to just give you a feeling for what the complexities are, what the challenges are, and how much we understand, and also to illustrate how we in neuroscience now try to approach a complex problem that still is very, very unclear on how to address it. So I will first describe social transmission of food preference and then tell you about these two studies. Okay, so let me explain what social transmission of food preference is. This talk is about food. Not dinner yet, but anyway. And when we talk about food, most of the time we talk about what's good food and what's bad food, and where to get good food and where to get bad food, or not to get bad food, right? So this is a very human thing, but mice do that too. They also tell each other about food. And the way this works is as follows. When you expose a mouse, a demonstrator mouse, to just plain food, and then you have it socially interact with an observer mouse, as shown here, and subsequently, outside, after the interaction, you give the observer mouse the choice between two different flavored foods, in this case cinnamon and cacao. The observer mouse generally will have a preference. They will like one type of food better than the other. As it turns out, at least at Stanford, observer mice, no matter what genetic strain, prefer cacao over cinnamon, as do I, by the way, just in case. That is the innate food preference. Now, if you then do a different experiment where you have the demonstrator mouse eat cinnamon-flavored food, then remove the food and then have it interact with an observer mouse. And you then give the observer mouse the choice. The preference is reversed. The demonstrator mouse tells the observer mouse in the social interaction that cinnamon food is just perfect to eat. It is the simultaneous olfactory and social stimulation in this interaction that causes this communication. Just to illustrate how this works, this is the innate food preference that I already mentioned. If we do social transmission of food preference training, STFP training, just as I explained it now, you can reverse this, and now cinnamon is preferred, this memory lasts for months. One interaction, the mouse will remember a month later what food is better, okay? Despite the fact that it would normally prefer cacao. It does require the social interaction because just having the mouse smell the cinnamon food doesn't work. Nor if you have a kind of a dummy, a mouse dummy that smells like cinnamon, doesn't work either. It really has to be a true social interaction. So the bottom line here is that STFP, social transmission of food preference memory, represents a one-trial appetitive, not aversive, learning paradigm. Very different from fear conditioning that many of us also use, which is aversive, also a one-trial learning paradigm. And it involves an ecologically relevant behavior because mice presumably will also tell each other in the wild what food is good or bad. What I want to do now in the subsequent half hour or so, I don't know how much time I have, 0, is to tell you about our attempts to understand this, how we do this, and what we find. And I will tell you two very different stories. One that was published some years ago, one that was just published a couple of months ago, about our successes and failures in trying to understand this. So the first story has as the starting question, how is the sensory odor information initially processed during STFP memory formation when the mice get together in the social context? And we hypothesized, obviously, that the olfactory sensory organ, olfactory bulb, is involved, and that feedback connections between the olfactory bulb and target projection areas must be feeding back into the olfactory bulb during the process. And so we looked at the synaptic connections of the olfactory bulb. You may remember from your neuroanatomy, if you ever had it, that the olfactory bulb has a main olfactory bulb that contains all the olfactory sensory inputs. And it, in addition, has an accessory olfactory bulb that senses pheromones. The main olfactory bulb contains multiple regions that it projects to. These projections are mediated by projection neurons called mitral cells and tufted cells, and they form axons and synaptic connections with the piriform cortex as the primary sensory cortex, and with the anterior olfactory nucleus, as shown here. These connections are bidirectional. There's also a feedback coming back. In addition, there are other connections, including to the ventral hippocampus. Two major reciprocal connections are thus involved in processing information from the main olfactory bulb to the central part of the brain. And we became interested in the AON because, as I tried to tell you earlier, we're interested in trying to understand at the molecular level how synapses are built. And it turns out that the AON uniquely expresses very high levels of a synaptogenic molecule called C1QL3. So let me explain this a bit. I showed you this slide already earlier. C1QLs are secreted C1q domain proteins. They contain a domain that's called a C1q domain that's also in complement factors, C1. And the C1QLs bind to BIs, which are adhesion GPCRs on the postsynaptic membrane. Because these molecules form a complex, and we know from previous work that we performed that C1QLs are synaptogenic, we hypothesized that C1QLs in the AON might be involved in projections to the olfactory bulb, and thereby regulate in some manner olfactory behaviors. And we intended to figure this out. What you see here schematically is how this complex works based on crystal and cryo-EM structures that we obtained, demonstrating that you have a postsynaptic adhesion GPCR called BI, a presynaptic adhesion molecule called RTN4R, and the secreted C1QLs, and they all together form a complex that somehow has to do with something, with synapses. So this is the molecule that is highly expressed in the AON. Now let me go a little bit into how we actually do such experiments when we are trying to understand circuits. What you see here is an experiment where we are mapping, using retrograde tracing, the synaptic connections between two different brain regions. And specifically, as you can see here, we, in this case, injected a retrograde tracer, choletoxin B, into the olfactory bulb. And then we identified brain regions to which this retrograde tracer would be transported. And we measured the extent to which the retrograde transporter, retrogradely transported tracer would overlap, in this case, with C1QL3 in expression. And this shows on the top left the injection site in the olfactory bulb. And you can see that when we do this, we find out that it's highly transported to the piriform cortex and to the AON. E and F shows the piriform cortex, C and D shows the AON, and you can also see that in the AON there's total, complete overlap with C1QL3 expressing neurons. So this raised then the question, given that there's this overlap, whether the C1QL3 molecule is essential for synaptic connections that are formed by the AON to the target molecules. We analyzed this using classical synaptotag tracing, which is another method in which we can trace synaptic connections, and using electrophysiology. I'm not going to go into the details here. I just want to point out that when you look at these slides, you can see that, oops, well, this is fancy, that here there's a decrease in puncta, decrease in synapses, and here in the input-output curve, or the slope measurements, there's a loss of synaptic connectivity. So this experiment, typical for the types of experiments we do, illustrates that these synapses depend on C1QL3, and thus C1QL3 truly is a synaptogenic molecule in this particular synaptic connection. So then comes the critical experiment. Does this connection have a role in STFP training? And if so, does it have a general role in all kinds of olfaction, or only in STFP training? The way we do this again is using stereotactic manipulations. We use the C1QL3 conditional knockout mice. We delete C1QL3 using these mice by injecting Cre or delta Cre as a control into the AON. Before training, then we train and we test. And you can see on the right that the deletion basically blocks STFP memory acquisition. In other words, you do need the retrograde connection for STFP memory acquisition. Great. This was done before we did the acquisition training. What happens if we do it afterwards, after the acquisition training, and then test longer-term memory? And what you see here is that if we do the injection immediately after training, and then measure later on, weeks later, STFP, it's normal. So if you do it afterwards, it's no longer required. It's only required for the training process. We then tested various other olfactory behaviors, just the ability to smell, a non-social olfactory memory. None of these were affected. This manipulation only affected STFP memory acquisition, and nothing else that we tested in our experiments, suggesting that the retrograde connection is selectively required for STFP memory acquisition, for the social component for social olfactory behavior, but not for other types of olfaction. Further experiments also showed that C1QL3 expression in AON to piriform cortex projecting neurons or to piriform cortex to olfactory bulb projecting neurons is not required. As I showed you earlier, the idea is that C1QL3 binds to a postsynaptic adhesion GPCR and thereby mediates synapse formation. We can test this by postsynaptically deleting the adhesion GPCR and performing the same experiments. What you see here is exactly such an experiment. When we do this now, deleting the postsynaptic adhesion GPCR, we also block the synaptic connection. And when we measure at STFP training, as you see here, same paradigm, we also block STFP memory acquisition, but we have no effect if we do the same experiment after memory acquisition, i.e. that connection again, shown here, is only required for the acquisition. Oops. So these experiments gave some insight into the acquisition process of STFP memory. They suggested that the AON has a critical role in this particular olfactory behavior paradigm, but only in doing the acquisition phase. They also showed a specific role for one particular pair of a synaptogen with its receptor in establishing a synaptic connection and maintaining the synaptic connections. In brain, synaptic connections very often turn over all the time. And so the maintenance of formation and the difference between formation and maintenance is actually not that clear. When we place this into the diagram of the memory formation phases that I showed you earlier, the sensory inputs operate via the main and the accessory olfactory bulb. Memory acquisition involves those sensory inputs, of course, in an interaction with the AON, with the piriform cortex, necessarily, because this is a contextual memory, and the ventral hippocampus. And this is mediated via this transsynaptic complex, because that is essential for this particular synaptic connection. Does that explain how memory acquisition works? No, it doesn't. But it does suggest that memory acquisition for this particular one-trial long-term learning involves a synaptic connection that is not required for other olfactory behaviors, which in some ways indicates that the initial interaction between the olfactory bulb and the AON is more complex during STFP memory acquisition than it is, for example, during normal just olfactory behaviors, normal smell behaviors. A specific transsynaptic signaling pathway underlies STFP memory acquisition, but is dispensable for later stages of long-term STFP memory or for other olfactory behaviors that we analyzed. So much for memory acquisition. Let me move on and tell you about the second project that I was going to discuss with you. And remind you that in mice, social signals are often, maybe mostly, communicated by olfactory inputs that operate via the accessory olfactory bulb. In fact, that is sort of the social nose of a mouse, and maybe of a human as well. So, this project starts again with a definition of circuits. And what I mean with that is the connections, the connectivity. Because despite all the connectome studies that are happening, we actually don't understand really the connectivity of the brain. In this case, we're again using a retrograde tracer, but we're using an AAV, a retro AAV. Details don't matter. It enables the same kind of experiment as I showed you before for the cholera toxin. And we analyzed the connections of the accessory olfactory bulb, not the main olfactory bulb, as I showed you earlier, with the brain. And when we did this, we surprisingly found that the major inputs to the accessory olfactory bulb from the central brain are in a somewhat enigmatic, weird brain region that is shown here, down here. And they're ipsilateral, or not contralateral. In addition, there's connections to the ventral hippocampus and to the entorhinal cortex. This brain region is the cortical amygdala. And specifically, it's not the entire cortical amygdala. It's part of the amygdala complex, which has more than 10 subnuclei. It is the posterior medial nucleus of the cortical amygdala, the COAPM, as shown here at the bottom. And quantifications demonstrate that in this quasi-cortical structure here, which is not a neocortex, but only 3-layered cortex, most of the connections are to layer 3 ipsilaterally. There's some to layer 2, only noise contralaterally. So this was a bit surprising because it suggested that there's a completely different cortical structure that projects to the AON, to the accessory olfactory bulb. And it led us to ask whether this might also have a specific role in STFP memory formation. So we did the same experiment that I showed you earlier, but now focused on the cortical amygdala. And what you see here is the experimental paradigm whereby 3 weeks before the training, we perform bilateral virus injections into the COAPM that express tetanus toxin to inactivate all outputs from the cortical amygdala, or GFP as a control. And then 3 weeks later, we train the mice, and then 3 weeks later, we test them. We also test them on the day of training, day 0, as shown here. And when we do this, as you can see here, or in the memory retention summary, GFP doesn't obviously do anything, it's the negative control, but tetanus toxin basically inactivates STFP learning only after 3 weeks, not on day 0. So it does not have any effect on memory acquisition, only on memory consolidation, storage, recall, reconsolidation, one of those. Okay? Consistent with this conclusion, if we do the same experiment, by using virus injections after STFP memory training, you get the same phenotype, the same impairment. So in other words, this structure, these connections from the cortical amygdala are required after training. They're not required during training, not during memory acquisition. Okay? So what time after training? When exactly does this happen? Okay? To test this further, as shown here, we specifically inactivated using AIV, retro AIVs with creoles, only those neurons in this cortical amygdala that project to the AOB. And we again impair memory training, either 3 weeks before or 1 day after. So it is those neurons that project to the AOB that are involved, essential for memory, long-term memory formation. Now this gives us an idea of how this might work, because it suggests that this particular structure is involved in some phase of long-term memory formation. But we don't know which phase. And in order to further tease this out, we used a technology that's called chemogenetics. This developed by Brian Roth at the University of North Carolina, where you can basically inactivate or activate neurons using a chemical agent in a defined time period. And the way this experiment was done is shown here. We inject into the AOB retroaccrete viruses and into the cortical amygdala the double-fluxed chemogenetic agent. I'm not going to go into the detail. What this does is that it selectively expresses the chemogenetic agent only in those neurons of the cortical amygdala that project to the AOB. And then we inactivate these neurons either in the first week after training or at the entire period before memory test or in the week before the memory test or just before the memory test, basically during the memory test, trying to test whether it's involved in the recall or in the consolidation. And as you can see here in the bottom, if we do it the entire 3 weeks, chemogenetics inactivates STFP, saline is a negative control. If we do it just before the test, either 40 minutes or during the 3rd week, just before this whole entire time, it has no effect. If we do it during the 1st week, it still impairs memory formation. So these experiments define a very short period when these neurons are required, the 1st week. Not during acquisition, not after the 1st week. In other words, they demonstrate that this particular brain structure is selectively required only for consolidation, not for storage, not for recall, not for acquisition. First time, I believe, that it's been possible to separate consolidation from storage and recall. One of the objections of the reviewers when we sent this in for publication was that many brain regions might have the same phenotype, that it's well known, which is true, that the brain operates by distributed information processing throughout the brain. And in this particular paradigm, STFP, there have been previous studies demonstrating convincingly that other brain regions are also involved. So this was a very reasonable objection that led us to test other brain regions as well. And what we found is if we did a similar experiment in the orbital frontal cortex as shown here, it does also impair memory, reproducing actually data, beautiful data, in the literature which already showed this, namely that the orbital frontal cortex, which has no connection with the olfactory bulb directly, only indirectly, is essential for STFP memory, long-term memory, although it turns out that it's essential for later stages. The ventral hippocampus, on the other hand, which is directly connected to both the main olfactory bulb and the accessory olfactory bulb, is not. There's no effect. Neither is the basolateral amygdala, another component of the amygdala complex, or the MPFC, the medial prefrontal cortex. So it is very selective. This process of memory formation, consolidation, doesn't just go everywhere. Now the problem here is, for those of you who study memory, that the ventral hippocampus, as part of the hippocampus, is supposed to be essential for contextual memories. In fact, whole careers are based on that. Nobel Prizes have been given for that. So clearly, this is a puzzling result, because here the ventral hippocampus is not essential. When we express, as shown here, tetanus toxin 1 day after training, how can we reconcile this? And so we tested with chemogenetics whether the ventral hippocampus might be required immediately after training, but not later. Because, as you probably all know, the ventral hippocampus is not really a hippocampus involved in long-term memory. It's more rather involved in short-term memory. And what you see here is that indeed, if we chemogenetically inactivate the ventral hippocampus 48 hours after training, it does impair STFP memory formation. At the same time, when we do this immediately after training for the COAPM, it has no effect. In other words, what happens is that consolidation does involve the ventral hippocampus at a phase after acquisition, where the information, the contextual information, needs to go through the hippocampus in order to be consolidated in the cortical amygdala, as shown here. In terms of the output pathways, it's actually unclear what brain regions the cortical amygdala communicates with. We did extensive circuit mapping and found a number of brain regions that are the targets of projections from the cortical amygdala, including, again, the AON, which turns out to be essential. Because when you, using, again, rather complicated technologies, inhibit those projections from the cortical amygdala to the AON, you can impair memory formation, long-term memory formation, not short-term memory formation. So this projection is, again, not required for acquisition, only for memory consolidation. And this is another approach for achieving the same thing, to just prove, as you can read in the paper, that it's really those synapses that are essential. So what does the STFP circuit look like now? We basically have these connections from the main olfactory bulb and from the accessory olfactory bulb that are then interconnected by synaptic connections between these various regions, project to the accessory olfactory nucleus, and then by an unknown pathway are communicated to the orbitofrontal cortex. Thus, we identified a circuit centered on the cortical amygdala that is selectively and only required for long-term STFP memory from consolidation. It is also a brain region, a circuit that involves protein synthesis in the cortical amygdala. How and why? So what does this truly selective consolidation process actually look like? What does it actually do? And so we use transcriptomics to explore gene expression mechanisms, because recent developments have enabled an unprecedented ability in the field to measure single-cell expression of mRNAs. The experimental strategy here is to use deep, smart single-cell RNA-seq. I don't know whether you're familiar with single-cell RNA-seq. There's different methods. There's drop-seq, this is very shallow. There's nuclear-seq, which is also very shallow. There's 10x genomics, which is intermediate. And then there's smart-seq, smart-2-seq, which is the only approach that gives you probably more than 50% of the mRNAs. Drop-seq probably gets you somewhere around 5 to 10%, so it's you're basically skimming the surface. The way how we designed this experiment is shown here again. We first label the projection neurons in the cortical amygdala that project to the AOB using a retro AAV. Then we do the behavior training. We have 3 conditions, either plain food, odor, or STFP training. 30 minutes later, we dissect the cortical amygdala and we do smart-seq 2. And when we do this, we define the neuronal composition of the cortical amygdala as shown here. There are 6 clusters in this U-mapped plot. Very simplified, that gives you an idea of a cortical structure like this that is basically composed of 6 principal types of neurons, which can be subdivided into subtypes. This is a unique cortical region distinct from other types of cortex. And what does STFP training do to gene expression here? This is a classical heat map that you've, I'm sure, seen before, and it compares the home gate condition, as shown here, with the odor condition and the STFP condition. And you can see that there is a series of genes that are selectively upregulated or downregulated as a function of STFP training, and that are absent from here, which are basically look like random noise. Among these genes that are upregulated by training RTN4Rs, the genes that I showed you earlier that encode the receptors in this bi-complex, for which C1QL3 is also a substrate. Multiple transsynaptic adhesion molecules, including RTN4Rs, that form a complex with bis and C1QLs, are identified here. To confirm these results and to compare STFP-induced gene expression changes between different brain regions, we performed spatial transcriptomics, MERFISH. As you can see here, same paradigm, exactly same experiments, except we're including the OFC, where we don't use labeling because it doesn't project to the accessory olfactory bulb. But instead of doing smart-seq, we do MERFISH, cryo-start sections. When you do this, you get these beautiful pictures of the brain. It's computationally very demanding. It shows you that the composition, the neuronal composition is quite different. What I find quite surprising and amazing, for example, if you look at the 6-layered cortex, it changes as we move along. For the cortex here, this is the hippocampus, and down here is the cortical amygdala. Here you can see the projection neurons that are TDP positive in layer 3 of the cortical amygdala. This is layer 2. And here you can see the olfactory bulb down here and the OFC above it. This enables us to test whether the smart-seq results are consistent with spatial analyses, and also makes it possible to compare different brain regions. And what we found in these experiments is that each brain region is different. And many brain regions really don't have specific changes as a function of STFP memory formation. What you see here is a comparison of the DEX between the cortical amygdala and ventral hippocampus. Both contain STFP-specific changes to some extent. But the ventral hippocampus genes do not overlap at all with the cortical amygdala. They're different. There are specific changes in gene expression programs that differ between the 2 different brain regions, especially with respect to the synaptic adhesion molecules as shown here. The same thing applies for the OFC, where there isn't even a change as a function of STFP memory formation. The change is entirely sensory, suggesting that the role of this region in STFP memory is contextual. It's basically identifying the sensation, not in the actual memory. And finally, if you compare if you look at which ones of these cortical amygdala specific tags are actually changed in the OFC, you can see there are none in STFP, although odor does something. So the comparison shows that the OFC, which is also essential for STFP memory consolidation in a protein synthesis dependent manner, senses odors, not social context. And the RTN4Rs lead us back to this complex that I indicated to you earlier, where we have these two different interactions that are involved both in memory acquisition and in memory consolidation. So the bottom line here is that the RTN4RL by 3 complex controls neural circuit formation by regulating various processes that are essential for synaptic connectivity that I'm not going to describe in detail, and that thereby they're critical for long-term memory formation. I'm at the end of my talk. I would like to summarize what I've tried to tell you here in the second part by coming back to this diagram, whereby the olfactory bulb obviously is essential for memory acquisition, and for memory acquisition specifically for the social memory, this involves retrograde connections from the AON, whereas the cortical amygdala is essential selectively for memory consolidation in a process that involves gene expression changes, which are required, even though the cortical amygdala itself is only required for a week. So that 1 week, gene expression changes are critical for memory consolidation. And in all of these processes, we believe there's evidence to think, although not yet compelling, that by 3 specifically mediated synapse formation is critical for all of these steps in different circuits. What is interesting here is that the cortical amygdala acts selectively only in memory consolidation, but requires gene expression changes. To the best of my knowledge, this is the first time a circuit has been shown to be only required for memory consolidation, and basically demonstrating that consolidation is truly separable from other components, at least of this type of long-term memory formation. How this actually works beyond, and why, for example, you need gene expression changes even for a transient stage in memory formation is unknown. So I'm ending here. I would like to acknowledge the people who did the work. The second part of my talk was done almost single-handedly by Jihui Lu, who now is back in China, where she has her own lab. The bio work that I didn't discuss in detail was done by Jia Wang and Yi Miao, who are about to leave the lab, form their own labs. The transcriptomics was done by Wenfei Sun, who is a joint postdoc with Steve Craig's lab at Stanford. It's been a great collaboration. And the work that I've discussed on the memory acquisition was done by Cosmos Bang, who is now a postdoc at Harvard University, and David Martinelli, who is now an assistant professor at the University of Connecticut. I've had many great collaborations, some of which are listed here, that have been instrumental for everything we have done. Thank you. Happy to discuss this or anything else you would like to discuss. Thank you. And you can see I didn't move out of this here. It's like a little, yeah. If I had known that this would be a red carpet, gee. Okay, anyway, yeah. Please, yes, I love questions.
So this wonderful neuron of yours that consolidates after a week. What happens in that neuron
If I knew that, I would be very happy.
Is it isolated from any further activities? Because it's
So
that
there
particular...
is a very popular concept in neuroscience, the engram concept, which basically says that you activate during memory acquisition a specific set of neurons that then encode the memory. You shake your head, which basically reflects my own feeling. I think it's a rather unsatisfactory concept at this point, especially since the memory that's being actually studied is always short-term memory. It's never long-term memory. And so the whole engram idea, which goes back more than 100 years, was really for long-term memory, but now people talk only about short-term memory. Yeah. We also refer to the cells that we label in an activity-dependent manner, not in this study. We published other papers on this as engram cells, but I find this concept rather unsatisfactory. Our hypothesis is that these gene expression changes, which happen in the majority of projecting neurons, that they change the neurons. But in electrophysiological studies that I haven't discussed here, we couldn't find a major change. We could find a small change in excitability, but not anything that we would really be excited about. What we would love to do is to map the synaptic connections quantitatively. We haven't done that because, as you might know, that's not that easy. Yeah. Please, go ahead. Yes.
Congratulations on your excellent presentation. Do we have any indication that during the consolidation period, some combinatorial and recombination capability is happening, which is the hallmark of creativity and innovation?
Okay, could you repeat that? I'm sorry. Yeah.
It's my secret accent. Is there any indication that during the consolidation period, we have some combination and recombination of stored and consolidated material, memory material, which is the hallmark of creativity and innovation?
Yeah, so the idea that we have, and this is hypothetical, based on the gene expression changes and based on basically hypothetical ideas of how memory might be encoded, that consolidation involves a change in synapses, and the output synapses from the cortical amygdala in this case. But the question is really, does that change involve a restructuring or formation of new synapses? I don't know. One thing, people always think we know a lot. We don't actually. We don't know how synapses are eliminated. We don't know how synapses are really formed. We know that synapses have a very highly dynamic nature that differs between different brain regions, but we also don't know how this is regulated. So the best evidence for the restructuring that we have comes from the gene expression changes that I showed you, which demonstrate that the major genes include, to a large part, genes that we and others have identified as being involved in synapse formation.
You find my question very naive, but it's coming from someone who studies Mendelian disorders and abnormalities in order to understand normality. I was wondering if there are mice, or human for that matter, that they only have long-term memory. And the reverse, if there are mice, humans, that have only short-term memory.
Yeah, great question. There's the famous case of HM in human history, in medical history. HM was a patient who, in the beginning of his 20s, was subject to a complete removal of his hippocampus and some of the temporal lobe. because he suffered from severe intractable epilepsy. It cured his epilepsy, but he was unable to learn anything new. He was unable to basically remember something that happened 5 minutes ago. But he had perfect memory of all experiences that happened before his operation. So he could draw a map of his childhood home. He could walk back to where he was as a child. He remembered everything. This experiment, which is unfortunate for the patient, I don't know if it's ethically a good or a bad thing, I have no idea, that's beyond my pay grade, but shows that you can actually separate long-term memory from the hippocampus, because the retrieval of the memory worked still, at least the explicit memories, right? Implicit memories actually still worked for the patient. So for example, he couldn't remember having met a person, but he could communicate implicitly, let's say, with a dog of that person. There are these stories of this patient. So in that's the best experiment I know, or best example I know, where you can actually separate long-term from short-term memory. In general, short-term memory is always a prerequisite for long-term memory, of course. But long-term memory, once it's stored, I think is independent of the hippocampus and can be separated if you eliminate the hippocampus afterwards.
That's quite an interesting one, because it's true that he had these bilateral hippocampal lesions and operations, I should say, and so on. But the trouble with his long-term memory, which everyone says was normal, there's a little grain of disbelief there, because if you look at patients, for example, with Alzheimer's disease, they lose their short-term memory very quickly, and they retain their long-term memory. Actually, if you measure the long-term memories, you give them cards or photographs of things that happened in their past, their long-term memory is changed, and is sometimes very badly changed, even though it seems that they remember things, they remember in the wrong order.
Yeah, I totally agree. There's two problems here. One is that long-term memory that requires a contextual recognition, of course, requires also the hippocampus. So if, for example, HM was asked to recognize something as opposed to explain something explicitly, his long-term memory would also be impaired. Yeah. The other thing, obviously, in Alzheimer's disease, as you all know, as a degeneration, it basically affects all neurons and it destroys the whole person, unfortunately.
I am surprised that you think that things happen in memory and in the brain that are not contextual.
Yeah.
What do you mean by that exactly? I mean, every time you do even the same thing again, day after day, the context changes, and yet you continue to do it well. For example, a tennis player will, who has a complete memory of how to play tennis that is subconscious in most cases, and when he thinks about it, it disintegrates. If he goes onto a new court, he doesn't change, but if he starts playing on a different terrain and so on, then we know that the contexts interfere with their games. You need think only of Nadal, who was excellent on the dry court, but when he came to Wimbledon, he used to have problems. So it seems to me that it must be very difficult to decide that a memory is non-contextual, completely non-contextual.
Yeah, maybe we're getting a bit into semantics. What I find interesting in the case of HM and similar patients is that they could verbally recapitulate memory. That's what I mean with non-contextual. So, in other words, it's not that they were put into a context, a sensory context, and then asked to recall the memory. It is that they were put into a cognitive context, if you want, okay, and asked to recall the memory. I think if they were put into a sensory context that is not explicit, like verbal, that they would have problems. Okay. I don't know that. It's an interesting question.
So your mouse experiments are controlled for time and sex and age and everything, so that you eliminate most of that sort of...
I'm sorry?
You eliminate most of the noise, and that's why you can repeat your experiments.
Yeah. Okay, somebody in the back?
Mr. Shukov, many congratulations about your speech. I have a question about the synapse genesis that happens when the mouse becomes older. When mouse become older, does something happen to all of those experiments and in the amygdala and the hippocampus synapses and all of that circular system when they become older?
I'm sorry, I didn't understand the question.
The question is more like what happens when, if we have your team have done the same experiments with older mice rather than younger mice.
Oh, the age. Yeah, great question. I don't know what would happen. Older mice are clearly not as good at learning than younger mice. I would suspect that they wouldn't be doing as well, but I can assure you that even when you reach my age, you can still learn. So it still works. So I don't know. Yeah.
Thank you very much.
Okay. Thank you. I think the next talk will be about a subject that is related to what I used to work on, which is secretion, but that's a long time ago.
Thank you, Professor. Thank you. Thank you. Thank you very much. And a minor present from us to you.
Thank you so much.
Thank you.
Wonderful.
Thank you.
Yeah? Okay. Now we start section 10, interactive workshop number 3 on internal medicine, endocrinology, and diabetes. I would like to welcome to the podium chairs Dr. Ioannis Zakiriazis, who will be online with us today, Dr. Andreas Ptaras, and Dr. Kati Ioannidou Stelia.
Ο κύριος Κυριαζής. Ωραία, να το. Και δεν θα κάνουμε και παρουσιάσεις, απλά τα ονόματα θα πούμε και στον τίτλο.
Αν θέλετε διαβάστε μόνο αυτά.
Ναι, αυτά τα ονόματα.
Και δεν θα έχει commentating.
Ναι, παρακαλούμε. Ναι, Γιάννη, προχώρησε.
Many thanks to the organizing committee for inviting me to participate in this excellent conference, and especially to the president and dear friend, Professor Ioannis Patrikios. At this roundtable titled Internal Medicine, Endocrinology, and Diabetes, the magnitude of an unresolved problem, advances, and new treatment approaches, my co-chairing fellows are Dr. Andreas Pitaras and Dr. Kadi Ioannidou Stelia. The first lecture we're about to listen has the title Revisit of the Dogma of Glucose-Stimulated Insulin Secretion in Pancreatic Beta Cells and News Beyond. As we all know, pancreas plays a critical role in maintaining glucose homeostasis through the secretion of hormones from the islet of Langerhans. Glucose-stimulated insulin secretion by the pancreatic B cells is the main mechanism for reducing elevated plasma glucose. Researchers have also uncovered a novel route to stimulate the growth of healthy insulin-producing pancreatic beta cells in a preclinical model of diabetes. The findings hold promise for future therapeutics that will improve the lives of individuals with type 2 diabetes. So, we have the privilege and pleasure to listen to the Professor's Wolfram Greiger speech regarding the specific topic. Professor Greiger, we are ready to listen to you. Is Professor Greiger listening?
Yeah, but it doesn't work here.
Μπορείτε να εμφανίσετε την παρουσίαση. Could you please... Okay. Could we see the presentation, please?
So meanwhile, I can tell you that I asked the organizer not to skip the break, so I have more listeners, but I failed, obviously. So, yeah, we can start. Thank you very much. So today what I'm going to tell you is, we switch gears from the brain to the pancreas, but it fits very well because beta cells are supposed to be also neuronal cells, so it fits very well to Professor Südler's talk. So what I'm going to tell you today is a little bit a rethinking about how beta cells work, and I think it's essential because we try to design therapeutic intervention against diabetes and how we prevent the progression of diabetes. I think when we try to do that, we should at least know how the beta cells work. And this is basically the common aim of beta cell knowledge. And I just go quickly through it, and I'm sure you know that so far we know that glucose uptake stimulates glycolysis, and glycolysis makes NADPH and pyruvate, and both NADPH and pyruvate enter the mitochondria, stimulating the cyclic cycle, again making NADH, and these both NADH fuel the OXPHOS, making ATP. ATP prevents the K-ATP channel, leading to depolarization. L-type channels go open, you get the calcium influx on vesicle fusion, and that's the mechanism for insulin secretion. However, there's one thing, and I tell you, working with mitochondria, it's always impressed me how people ignore that, because the one thing is the mitochondrial ATP production, as we know, depends on the OXPHOS substrate, entirely NADH and FADH, and also an activity of citric cycle, which produces the NADH again. So in other words, to get the OXPHOS activation and the ATP production, the glycolysis-derived NADH has to enter the mitochondria, and that is mastered by a protein that's called ARALA. And ARALA is one of the calcium-dependent part of the malate-aspartate shuttle. On the other side, citric cycling on the other side is also a calcium-dependent mechanism, in particular the isocitrate dehydrogenase is essential dependent on calcium. As you see here, there are two main mechanisms that drives high glucose-induced ATP formation are dependent on calcium. However, the model doesn't indicate calcium being involved at all. So in other words, when ARLA and EDH are not activated by calcium, the glycolysis would be disconnected from mitochondrial ATP production, and the beta cell wouldn't be able to sense glucose elevation. So in this regard, the beta cell have a very specific mechanism to ensure the direct coupling between the glycolysis with the OX-phos ATP production. And it is by the beta cells produce a very unique calcium leak from the endoplasmatic reticulum towards the mitochondria, and that is established by presenilin 1. And I hope you realize now, listening to presenilin 1, that really beta cells are somehow familiar to neurons, because we all know that presenilin 1 pops up on Alzheimer's disease. So what is about this presenilin 1 mediated calcium leak? So the mediated calcium leak goes directly to mitochondria. I try to use that. Yeah, here. So the presenilin 1 here, together with some helper proteins, I don't want to go in detail here, releases calcium continuously towards the mitochondria. And you have to realize that the ER calcium is 4,000 times higher than the cytosol. So once when you put a small leak, there's a lot of calcium going through, and this calcium is directed entirely towards the mitochondria. And there, it obviously activates, it does exactly what I told you before. It stimulates ARLA and stimulates the TCA cycle. And the mechanism on the other side needs to be equipped that this calcium that comes from the ER has to be refilled, compensated, or refilled continuously from the extracellular space. And this mechanism has been found by a sodium-calcium exchanger here, and then so far non-found, non-discovered sodium channel and TRPC3. But mainly the sodium channel allows sodium to enter the beta cell, and this will stimulate the NCX1, and NCX1 shuffles out sodium again and calcium in. And this calcium is reuptaken by the ER. And as you can see, these mechanisms so far completely unexplored as therapeutic targets on one side, but also a mechanism to introduce diabetes. And what I will show you in the next few minutes is how these mechanisms are important to develop diabetes, and maybe these mechanisms are the clue to understand how type 1 diabetes occurs so suddenly. Okay, so just coming back, what does presenilin, how important presenilin 1 is, a very simple experiment. This is a control, and if you have a beta cell and you expose a beta cell to high glucose, it starts oscillating in calcium, meaning, so now the beta cell senses the glucose, now it can release insulin. On the other side, when you knock down presenilin 1, you see that these oscillations are pretty much reduced, and it results in the reduction of insulin secretion. And I hope you appreciate here the effect of this, all that the presenilin will knock down has particularly the initial insulin secretion is completely wiped out, while a long-lasting insulin secretion remains. And this particular first insulin secretion is very important for tissue priming, and we think about this as a pre-diabetic state. Okay, so in other words, by organizing mitochondrial-oriented calcium leak from the ER towards the mitochondria, presenilin 1 establishes the calcium hotspots in the so-called mitochondria ER calcium contact sites called shorter MERCs. There, this high calcium elevates matrix calcium, stimulates ARLA, and yields a pre-activation of mitochondria despite there's no high glucose. So in other words, if you are sedentary, still your mitochondria is pre-activated in the beta cell, although there's no substrate, but the calcium is high. And the mitochondria basically waits that substrates come and then is producing ATP. So this we call metabolic priming. And in other words, we found out in another stories that this metabolic priming ensures the beta cell susceptibility to elevated glucose. But today I just want to do one step further. And this one step is that we have recently found that this metabolic priming that occurs in the gap between the ER and the mitochondria by increasing calcium and stimulating the uptake of NADH, meaning recycling NADH to NAD, is extremely important to run glycolysis per se. And when you knock down presenilin-1, and this is a very, I think, fundamental finding, beta cells stop making glycolysis. So in other words, when you don't have presenilin-1 here, the beta cell glycolysis makes a bottleneck, which is the first step that needs NADH, because here NADH is not existing anymore, cannot recycle because RLI is not activated. Consequently, the glycolysis is not finished. So a beta cell that has no presenilin-1 or a dysfunctional presenilin-1 doesn't have a glycolysis anymore. And the intermediates of the upper part of the glycolysis, these obviously, and this was our idea, might try to trigger a loss of beta cell integrity. So not only that you don't sense glucose, we explored whether these conditions also sensing the lack of identity of beta cell getting maybe an explanation of diabetes. So what we tested is some beta cell marker, and as you appreciate here is that the lack of presenilin-1 in INS cells reduced all specific beta cell differentiation markers, but also in human beta cell cells, you see a decrease in beta cell markers. And finally, we had also the same experiments in mouse pancreatic slices, and you can see here that a knockdown of presenilin-1 yields a decrease of beta cell differentiation markers. So in other words, presenilin-1 not only prevents glucose sensing, it also yields the loss of identity of the beta cell. So what we did is now we tested whether we can rescue when we reduce the glucose exposure of the cells. And as you can see here, just decreasing glucose concentration, you see a normalization of the signal. And this is meaning again that these intermediates of the glycolysis that are accumulating during this in the absence of presenilin-1 yields a normalization of this procedure to a normal beta cell. Interestingly, and this is again a link to the previous talk, we found the same phenotype in an Alzheimer's disease mouse. So what we did is we took the famous 3xTG Alzheimer's disease mice and tested the same protocols in the islets, and you can see here there is a small decrease of several markers of beta cell markers, and interestingly also insulin secretion, particularly the first fast insulin secretion, was drastically reduced, while a long-lasting decrease in insulin secretion was gone. So in other words, here we have discovered a new common molecular mechanism between the Alzheimer's disease and the beta cell dysfunction, and type 1 diabetes. So this, I make the story very short. So we tested all these molecular mechanisms, and you know, I'm a molecular biologist, so we like to play around with these DC tools. But we finally had a nice idea what signal transduction means here. But, and most importantly, we had an effect that AMP kinase, which is a very important sensor of starvation, was decreased while mTOR was increased. So there was a lot of signaling. However, and this was very surprising for us, no intervention against AMP kinase or mTOR could rescue the beta cells of losing its identity. So we were thinking, you know, this beta cell on one side is losing its identity, on the other side it has a lot of signaling molecules changing, but they didn't have an effect. So we're wondering what basically was the effect, and this is a very short slide because I'm run out of time. What we find out is that particularly these signaling molecules are important to normalize the beta cell function in terms of immunogenicity. So when you don't have this signaling by presenilin 1, suddenly PD-L1, which is a checkpoint protein on the surface of the beta cells, to kind of calm down the immune system. And now we had suddenly this PD-L1 strongly reduced by presenilin 1 knockdown, meaning that this beta cell is very susceptible to immune system, and once the immune system gets activated, by example, measles or some other virus attack, this might result in an attack of the beta cells by the immune system. So just to summarize, this is the normal signaling pathways, how a beta cell on one side saves its integrity and also its functionality. On the other side, when you don't have presenilin 1 that makes this ER calcium leak in the mitochondrial ER calcium contact sites, you suddenly have no glycolysis anymore, but no intact glycolysis, but high glycolytic intermediates, because suddenly you have this bottleneck function. And this leads to loss of identity of the beta cells. On the other side, you have a signaling cascades, particular various different activation of calcineurin, MFAT, AMP kinase, and mTOR that leads to beta cell dysfunction. And overall, I hope I can show you now two new approaches. On one side, we can protect really from diabetes because we can fight against the loss of beta cell identity. And I'm very happy to tell you that the first trials already running in our institution that we try to really prolong the onset of diabetes in human. On the other side, we can intervene the beta cell dysfunction, maybe helping people with already existing diabetes mellitus. With that, I would like to acknowledge my team, particularly the guys who are in reddish, that have mainly contributed to this team. And I thank you for your attention. Thank you very much.
Thank you very much. We don't have any questions because we don't have time. Sorry, we don't need questions. So I'll call the next speaker, Professor Hadji Gelaki, dealing a lecture entitled Looking for the Future, From Clinical to Precision Medicine.
Dear chairs, dear colleagues, first of all, I would like to thank the organizing committee and especially my friend, Professor Ioannis Patrikios, for this kind invitation. As you can see, my talk is about a look to the future, from clinical medicine to precision medicine. On this slide, you can see the shape of the Earth from the space in the early 60s, which coincided with the approval of metformin. However, today, this image of the Earth from the space looks completely different. And this circular shape of the Earth is depicted in the consensus for the holistic treatment of type 2 diabetes, and also as the cover page of the supplement of the Diabetologia journal referring to precision diabetes medicine. But what is actually precision medicine? It's an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. And this approach allows all clinicians and researchers to predict more accurately which treatment and prevention strategies will work for a particular disease and in which groups of people. It is in contrast to a one-size-fits-all approach. But starting with phenotype, the word phenotype is used with many different meanings, and in the biology, the most widely accepted definition of phenotype is observable traits of an organism. And deep phenotyping is defined as the precise and comprehensive analysis of phenotypic abnormalities, often for the purpose of scientific examination of human disease. And why should we care about this precise analysis of human phenotype? Because the combination of deep phenotype data with an increasing amounts of genomic data appear to have an enormous potential to accelerate the identification of complications of disease subtypes with prognostic or therapeutic implications. Are we actually ready for such a project? As the medical field has embraced the omics area and big data have provided novel insights into pathophysiology and disease progression, the term precision medicine has been coined to capture the promise of tailoring. It is a movement from one-size-fits-all to personalized therapy. Additionally, precision medicine has also the potential to be used for precision prevention. There is a consensus from the American and the European Diabetes Association about precision medicine in diabetes. And the purpose of this consensus is to define relevant terminology and review the current status of diagnostics and therapeutics in diabetes. Particular focus is placed on elucidating the etiological heterogeneity of diabetes, as well as lifestyle and pharmacological interventions. According to this consensus, diagnosis involves refining the characterization of the diabetes diagnosis for therapeutic optimization or to improve prognostic clarity using information about a person's unique biology. And precision diagnostics employs methods to subclassify patients to enable the successful application of precision medicine approaches. However, the precision treatment the target of precision treatment is to optimize therapy in order to achieve therapeutic goals and to reduce the side effects. Precision therapeutics involve tailored treatment based on information about an individual's unique biology, and precision prevention determines likely responses to health interventions and risk factors, optimizes also interventions and/or minimizes risk factor exposures for a given individual. Monitoring is also valuable for prevention, diagnosis, and prognosis, and includes detailed assessments of biological markers, behavior, diet, sleep, and psychophysiological stress. The focus of precision prognostics includes predicting the risk and severity of diabetes complications, patient-centered outcomes, and/or early mortality, and we have to take into account the clinical and environmental characteristics. The path to precision medicine, to precision diabetes medicine, is a long one. First of all, we have to understand the disease. To take into account all the biomarkers and algorithms, clinician education is needed to take into account also the health economics and, very important, the patient engagement and the feedback of the patient. In addition to all this, there is also the omics universe. Emerging omics technologies such as proteomics, transcriptomics, and many other omics might provide new opportunities in understanding the underlying molecular disease mechanisms, enabling the design of new effective therapies. However, no single omics data type is able to reveal the complex behavior of cells or tissues in different states. There are biological networks that can be used for integration of multiomics data, and this approach might reveal phenotypic differences and underlying molecular mechanisms involved in the occurrence of the disease. According to Einstein, in theory and practice are the same. In practice, they are not. Now, how close are we to precision medicine? We know that there is an alarming increase in the prevalence of diabetes, and is estimated to affect almost 700 million adults by 2050. So a potential solution to reduce the rising diabetes expenditures and reduce inequalities in diabetes care is to shift toward precision medicine. The most recognized classification of diabetes is that from the American Diabetes Association and from the WHO. And nowadays, we have new approaches to precision diagnosis with newer conceptual models. Such a new conceptual classification model subtypes diabetes into 5 distinct clusters and is based on 6 key variables, such as gut antibodies, age of diagnosis, BMI, HbA1c, beta cell function, and insulin resistance. Each cluster has distinctive treatment pathways and varying complication risk assessment profiles to diagnosis. This Swedish registry, the ENDIS registry, was actually a great stimulus for further arising interest on the complex phenotypes of diabetes, and according to this, we have now these 5 new subclasses of diabetes: the severe autoimmune diabetes, the severe insulin-deficient diabetes, the severe insulin-resistant diabetes, the mild obesity-related, and the mild age-related diabetes. The rationale for this diabetes classification was based on the observation that people with diabetes have different phenotypes and show also a broad variation in the main features of diabetes. According to this classification, complications can be predicted much better than before. And we can decide and choose the right medication for the right patient in order to avoid also the complications. Additionally, precision medicine utilizes genetics. There are more than 400 genetic variants that have been identified, and more research is needed to make precision medicine a valuable option for all types of diabetes. And we have also to adapt our interventions concerning the treatment of diabetes. A few years ago, this article pointed out an urgent need for the review of the current diabetes classification, and also a need to align the classification in line with the known etiology and disease course. And the proposed model was the beta cell-centric classification of diabetes. This beta cell-centric classification system supports the interplay of genetics, insulin resistance, environmental factors, and inflammation in the immune system that cause beta cell disruptions. This beta cell-centric model recognizes 11 interlocking pathways that contribute to hyperglycemia, as you can see on this slide. And on the right, you can see the mediating pathways of hyperglycemia addressed by various available agents. The optimal strategy is to use the least number of agents to target the greatest number of mediating pathways of hyperglycemia. However, this is not enough. Pharmacogenomics is essential for the application of precision medicine, and what is it is one of the emerging approaches to precision medicine, tailoring drug selection and dosing to the patient's genetic features. What we know nowadays about the new drug classes we have available on the market, there are some genotypes that are associated with much better response to treatment of type 2 diabetes using the new drug classes. And this very interesting article pointed out that there are 146 targets of SGLT2 inhibitors that intersected with the disease. And on the right, you see an interaction network for drug target discovery consisting of SGLT2 inhibitors and these 146 targets. Now to the question, how far are we from precision medicine? While precision medicine has a potential to be a game changer, its application is not without challenges. Challenges in diagnosis and classification, challenges in prevention, challenges in monitoring and treatment. Some of these challenges pertain to lack of data. Others are linked to inequalities in the availability and affordability of various diagnostic and therapeutic modalities. It is to be hoped that further advances in this field will bring personalized care within the reach of every individual with diabetes. Now, coming to the end of my talk, we knew genotype, phenotype, and clinical features. Biomarker profile, mediators profile, and omics have been added, and this composition has led from the old classification to the different outcomes and cardiovascular risk factors to composite genetic architecture and to different metabolic profiles. Led also from new technology to better understanding of metabolic pathways and from large databases to patient networks. And all this led to improved care and outcomes. However, dear colleagues, the future of personalized healthcare lies not only in prevention, in precision medicine, but also in artificial intelligence. Both impact the goal of personalizing care in 5 different ways, and their impact is consistent with the ultimate goal of prevention and early detection of diseases. This could actually reduce the burden of disease on the general public and thus the cost of preventive health care for all. And we have learned much about precision medicine, and we will continue to learn. However, its future is complicated, extremely complicated, I would say, like a maze. Probably one of you can find at the end the way to the exit. So, putting all this data of precision medicine in the center of discussion, diabetes seems to be treated in a much more charming manner than before. And with this, I would like to thank you for your attention. Now, I would like to please to upload my questions. Okay, you can scan the QR code and then vote, if it's possible. The question is, what is the role of precision medicine? Right. Oh, 50-50. 30-30. One and two, correct. Right. The next one, please. Classification of diabetes is based on the following key variables. Please vote. The right answer is the A. Gut antibodies, HO diagnosed BMI, HbA1c, beta cell function, and insulin resistance. And the last one, please. Common features between severe autoimmune diabetes and severe insulin-deficient diabetes. Please vote. E is not correct. The correct answer is the A. Early onset diabetes, low BMI, high HbA1c, and insulin deficiency. So thank you very much for your attention.
Thank you very much for an excellent presentation. Due to shortage of time, we will continue with our last presentation. And I would now like to present my co-chair, Professor Andreas Pitaras, who's a clinical hypertension specialist at George Washington University and adjunct professor of the School of Medicine at the European University of Cyprus. Thank you.
Dear Chair, ladies and gentlemen, we all know that lifestyle changes is the hard homework for the patient. So as you can see here, it's not so popular. And all the international societies suggest lifestyle interventions from the beginning of every step in hypertension management. Here you can see the European Society of Hypertension guidelines, and here the International Society of Hypertension in collaboration with European Society of Hypertension. You can see here four fields: movement and body weight, food and drink, body and mind, and others. And recently, the European Society of Cardiology guidelines here suggest many lifestyle changes in order to get blood pressure reduction and cardiovascular risk reduction. Let's start with exercise, which has a therapeutic, prognostic, and preventing role in the management of hypertension, and has a beneficial effect in every single risk factor and disease, including hypertension, reducing the systolic and diastolic blood pressure by 4 to 10 millimeters and 3 to 8 millimeters respectively. And here in an old publication in New England Journal of Medicine, following patients with severe hypertension taking at least 4 different drugs and having left ventricular hypertrophy, as you can see here, a clear reduction in blood pressure, reducing the medication up to 33%, and we saw a regression in LVH, in left ventricular hypertrophy. But the most difficult exercise is to go to the gym. So we need to improve the implementation of exercise guidelines, because the different exercise programs typically report dropout rates up to 45%. And if we include the wives in the exercise programs, like a typical Greek mother-in-law, the benefits are greater. Unfortunately, only 1 of 3 physicians advise an exercise program to their patients. And the message is that rather than canceling more than 30 minutes per day, we can recommend just 10 minutes of moderate intensity exercise for 3 days per week, because a body at rest tends to remain at rest, whereas a body in motion tends to remain in motion, so we simply need to get patients to act. We need to give a real exercise prescription in every single patient and include exercise into our treatment plans. The European guidelines suggest up to 300 minutes of aerobic exercise per week of moderate intensity, or alternatively, up to 150 minutes per week of aerobic exercise of vigorous intensity, and supplemented with dynamic resistant exercise. The European Society of Cardiology guidelines suggest more than 150 minutes per week, 30 minutes for 5 to 7 days per week. And here, the International Society of Hypertension suggests all these things for patients. We need at least 3 meds, and you don't have to be a marathon runner to get all these benefits. Just 5 to 10 meds is enough to get all these benefits. And we published recently 2 papers in JACC, which rated as the most important publication for the JAC for the years 2022 and 2023. Here, the first one, the higher the exercise capacity, the lower the mortality rates here in 750,000 population. And you can see here, even in octogenarians, the benefit is there. The higher the exercise capacity, the lower the mortality. And we saw in the other publication that even 1 or 2 met increase in exercise capacity, we have a benefit with decrease in mortality. And on the other hand, a decrease in exercise capacity, we can see a harm increasing the mortality. We all know that in real world, we have many barriers to physical activity, and we have to give many solutions to overcome those barriers to physical activity. And to say that there are 1,440 minutes in each day, and just spend 30 of these minutes doing something that is physically challenging, to follow the activity pyramid, and with no excuses. Going to the diet, beyond the religious approach, swallow your pride, it contains no calories, the guidelines suggest more plant-based and less animal-based food, salt substitutes with sodium chloride, except in CKD, in renal dysfunction, salt restriction, less than 5 grams, about 2 grams of sodium or teaspoon, increased potassium, except of patients with advanced CKD. Another guidelines approach is reduction to alcohol with a strong suggestion, and adopting a healthy and balanced diet such as Mediterranean or DAS diets. Drink less alcohol and restrict free sugar consumption and sugar-sweetened beverages. And the ISA-GSA guidelines suggest, as you can see here, the fruits and vegetables, protein, potassium, and so forth. Because our food is the #1 cause of poor health. And we faced recently a fast COVID pandemic on top of diet-related diseases as diabetes, obesity, hypertension, worsening outcomes as we all know. And about the nutrition, there is both passion and confusion. We have to prefer healthy foods and not isolated nutrients, and to choose quality carbohydrates with a low glycemic response, and to avoid processed meat because it increases the relative risk, as you can see here, more than 2 times. And to prefer all the protective foods. And we have to know that just small changes can have a big impact. Here is the Mediterranean diet, with some variations depend on the different countries. And this is a typical modern diet pyramid, DAS diet. The salt is pretty important in the management of hypertension. Here, the quantity of sodium in DAS diet. And we have to look for the hidden salt in processed foods. It's pretty important. And you can see here the clear difference between unprocessed and processed foods. You can see the clear difference in salt here. Going to the mind-heart-body connection, we all know that stress has a strong relationship with blood pressure, and we all know the Takotsubo stress cardiomyopathy. And the guidelines suggest reduced stress via controlled breathing exercises, mindfulness-based exercise and meditation. And here, good sleep, meditation or yoga, and listen to music. To have a possible benefit on cardiovascular risk. And here are 7 ways to reduce stress, as you can see here. This is a nice banner to have to our office and to see it and to see the messages. And the most important thing is that food is the most abused anxiety drug. On the other hand, exercise is the most underutilized antidepressant. Going to the smoking, smoking cessation and referral to smoking cessation programs to avoid blood pressure increases, to reduce the risk of masked hypertension and improve cardiovascular health outcomes. You can see here on the other guidelines, the International Society of Hypertension, and here the European Society of Cardiology, the same approach. But unfortunately, patients enjoy smoking. They usually know the risk of smoking. Patients are not motivated and they have too much stress. We have to inform our patients that smoking leads to up to 11 years loss of lifespan. And here is a nice table from the European Prevention Guidelines showing the lifetime gain from smoking cessation for every single patient. Here, for an example, 3 years. We can give some therapies for smoking cessation, and we need to allow to delay quit date after starting medication, because 40% of smokers make a quit attempt each year. 44 want to quit gradually, and developing effective gradual reduction could engage more smokers in quitting. And we need to inform our patients that there is no safe level of smoking. Even 5 cigarettes per day indicate a higher risk. And we have to have a clear schedule in order to stop smoking. And here to indicate all the benefits of smoking cessation. Another good idea is a mobile cardiometabolic cardiorespiratory unit to have a clear benefit and to give a holistic point of view. Obesity gives a hard time to the cardiovascular system, especially for the younger, and the guidelines give information for weight reduction. Here to maintain healthy weight and waist to height ratio less than 0.5, and keeping the BMI between 20 and 25 kilograms per square meter, and waist less than 94 centimeters in men and less than 80 centimeters in women. Unfortunately, there is a delay to ask for medical advice up to 11 years. And we have the approach of 5 A's: ask, assess, advise, agree, and assist. And you can see here the current options in obesity management, the lifestyle changes, medication, endoscopy procedures, and the bariatric surgery. Here are the three pillars: psychological intervention, pharmacological, bariatric surgery, and on top the physical activity, medical adherence, and medical nutrition therapy. The most important thing is that it's not finding the time you think you need, it's using the time that you have and always ask for permission. It's very important to repeat the messages and to see the patient as equal partner, sharing the patient needs and wants and clinician knowledge and recommendation. Motivation makes you start, habit makes you continue. If it's important for you, you will find a way to do it. If it's not, you will find an excuse. And usually we say, yesterday you said tomorrow. And if you are persistent, you will get it. And if you are consistent, you will keep it. And the promise of prevention depends on robust healthcare delivery. And it's never too late. Thank you very much for your attention.
Thank you very much. Apologies to the audience and to the previous speaker. There is a hybrid last lecture by Professor Makaritsis, who is a professor of internal medicine at the University of Thessaloniki Medical School in Larissa, Greece. Yes, we can hear you if you want to make a start.
Can you see the slides, please? Okay. Well, good evening, everyone, dear colleagues. Because of an unforeseen illness, I could not be there with you. First of all, I would like to thank the organizing committee and especially Professor Patricios for this honorary invitation again to be part of this spectacular congress. And give me a minute. Well, my lecture is about the diagnostic and therapeutic approach to a patient with hypernatremia. We're going to discuss about an 80-year-old male patient that has a body weight of 70 kilograms with a history of dementia, who was admitted to the hospital because of liver somnolence, diminished fluid ingestion, and oliguria of 5 days duration. On clinical examination, the patient was febrile with dry mucus in brains, decreased skin turgor, and tachypnea, and the blood pressure was 85/50 millimeters of mercury, while the patient was in the supine position, and the pulse was 125 beats per minute. Lab tests showed leukocytosis, increased CRP levels, increased BUN levels, creatinine out of normal range, glucose 140 milligrams per deciliter, low potassium 3.1 millimoles per liter, but an extremely high sodium of 160 millimoles per liter. In this patient, the central venous catheter was placed into the right jugular vein, and the central venous pressure was measured very low at 1 centimeter of water. Now going back to a little physiology, with regard to plasma osmolality, we know that osmolality of solution, the best method to be measured is directly by osmometer, but this is not always feasible. And for this reason, there are several empirical formulas to calculate plasma osmolality. The best known is the Worth-Rate equation, by which we can calculate plasma osmolality. This is the equation of the sodium concentration multiplied by 2, plus the glucose concentration divided by T, and concentration divided by 2.8. And we all know that plasma sodium concentration is the primary determinant of the plasma osmolality. It can be approximately expressed that plasma osmolality is 2 times the sodium concentration in millimoles per liter. Factor 2, we know, is used to account for the anions that accompany sodium in the extracellular space, that is chloride and bicarbonate. Now, with regard to the definition of hypernatremia, we know that it's a rise of sodium concentration higher than 145. Plasma sodium is an impermeable solid because of the action of potassium sodium ATPase, and it contributes to tonicity and can induce actually the movement of water across cell membranes. Therefore, hypernatremia always denotes hypertonic hyperosmolarity and always causes cellular dehydration because water moves from hyposmolar to hyperosmolar areas. Clinically, hypernatremia is divided into acute, that develops within 48 hours, and chronic one, that develops more than 48 hours, or if there is unknown the time of initiation. And because sustained hypernatremia can actually occur only when thirst perception or water access is impaired, the groups at higher risk of getting hypernatremia are patients with altered mental status, intubated patients, infants, and of course, elderly persons. Please upload the first question, question number one. Question number one is, which of the following statements about hypernatremia is false? I don't know if I have to wait for the audience to vote. A, it is a state of isoosmolality, results in intracellular fluid volume contraction, stimulates thirst, excretion of minimum volume of maximally concentrated urine. It is commonly caused by insufficient water intake. We are looking for the false statement. Please let me know if I have to proceed or wait. Are you there?
If you could scan the QR code and respond to the question, please.
So the correct answer is that it's a state of isoosmolality. Actually, the false statement is the correct answer. We have already said that hypernatremia. Now, a few words about epidemiology. This is a common electrolyte disorder, an incident of 1 to higher than 3%. But mortality rates are very significant, from 40 to 60%. And the highest mortality is seen in patients with hospital-acquired hypernatremia. One important thing here is that the majority of hypernatremic patients on admission exhibit also hypovolemia. There is a low volume, it's a low volume state, and half also of these patients have at least one additional electrolyte disturbance. So a lab test for the other electrolytes should be done. Hypernatremia is also particularly common in patients in intensive care units, from 7 to 16%, as you can see, and also in about 8% of neurosurgical intensive care units. A little in pathophysiology, when we have hypernatremia, we said there is an increase in plasma osmolarity, so that actually stimulates the osmoreceptors located at the hypothalamus, and this has two responses: the release of an antidiuretic hormone, which actually acts on the collecting tubule in order to increase the reabsorption of free water. And secondly, it's the thirst stimulation that increases the water intake by mouth from the patient. And these two actions, these two responses actually try to restore plasma osmolality back to normal. With regard to the clinical types, the acute type, as we said, is when that develops in less than 48 hours. There is an intracellular dehydration. As I said before, there's a movement of water from the cells to the plasma, and that leads to shrinking of brave cells. And this is most probably the reason of the neurologic symptoms of acute hypernatremia. But when there is a chronic initiation, more than 48 hours, it happens an adaptive response with adaptive retention of solids into the brain cells, in order to have restoration of the brain cell volume. And symptoms there can develop, are mild. They can develop when iatrogenically we try to get a rapid correction of the hypertonicity. In that case, we might get cerebral edema. Now, with regard to the causes of hypernatremia, hypernatremia results either from net water loss, but there is no sodium loss together. And that happens, of course, in diabetes insipidus, either neurogenic or nephrogenic. Drugs that can also cause most probably nephrogenic diabetes insipidus, like lithium, amphotericin B, or valproate, in cases of hypercalcemia or hypokalemia. The second cause is to have hypotonic fluid loss, meaning that you have loss of water, but this loss of water is higher than sodium and potassium losses together. That happens in renal, like loop or osmotic diuretics, in gastrointestinal tract losses, vomiting, diarrhea, or lactulose medication, or skin from cutaneous losses from burns or excessive sweating. More rarely, it might happen also from hypertonic sodium administration, and that is mainly iatrogenic. Please upload the question number 2. Question number 2 is, which of the following types of hypernatremia occurs when sodium loss is accompanied by a relatively greater loss of water from the body? Is it essential, euvolemic, hypervolemic, hypovolemic, or none of the above? Actually, what we just said about that there is a greater loss of water compared to the sodium or potassium losses. That results in which type of hypernatremia? Tell me when we proceed, please. Panel. Well, the correct answer is B. In these cases, we have hypovolemia. Actually, as we said before, this is the most common type of hypernatremia. Now, with regard to clinical manifestations and diagnosis of hypernatremia, of course, history, clinical examination, and lab determine the cause of hypernatremia. It's very important to determine the cause. And the increased sodium levels, especially in acute hypernatremia, because of the, as we said, the hydration of the brain cells, results in lethargy, weakness, irritability, seizures, and coma. But this also brain shrinkage induced by acute hypernatremia can cause vascular rupture, bleeding, subarachnoid, and death. That is the cause of death. This is the cause of death in patients with hypernatremia. On the contrary, in chronic hypernatremia, brain shrinkage is countered, as we said before, by an adaptive response, which promotes the accumulation of organic osmolytes like glutamate, taurine, and myoinositol. And in this way, there is a pooling of water into the cell, trying to restore the cell volume, the brain cell volume, and that accounts for the milder symptoms of hypernatremia that develop slowly. So let's move to the last part, is the management of hypernatremia. To get a detailed medical history is very important in order to address the underlying cause. The second most important thing is to assess the extracellular volume status, actually the effective arterial volume, and I will explain why. And third, it comes third to correct the hypertonicity. In order to manage the underlying cause, we have to stop any fluid loss from the gastrointestinal, from renal, control pyrexia or hypoglycemia, to stop lactulose and other diuretics, to treat if there is any hypercalcemia or hypokalemia, and to moderate medication like lithium or other medication that can cause polyuria. Now, there is a difference between the management of acute and chronic hypernatremia. In cases of acute hypernatremia, it has been shown that rapid correction improves the prognosis without increasing the risk of cerebral edema, because the accumulated electrolytes are rapidly extruded from brain cells. So in these patients, reducing the serum sodium concentration by 1 millimoles per liter per hour for the first 6 to 8 hours is appropriate, in order actually to prevent the consequences we said before, the cellular dehydration, which might induce intracerebral or subarachnoid hemorrhage. This is not the case in the management of chronic hypernatremia. In patients with chronic hypernatremia that develops in longer than 48 hours or it's of unknown duration, the reduction of the sodium should be done very slowly at a maximal rate of 0.5 millimoles per liter per hour in order to prevent the cerebral edema and convulsion. It is recommended a targeted fall in the serum sodium of less than 8 to 10 millimoles per liter per day. The goal is to reduce sodium to 145, and in cases that patients develop seizures, they might need prompt anticonvulsant therapy or adequate ventilation. How can we calculate the water deficit? The calculated water deficit is calculated by this formula in order to get, as we said, levels of less than 8 to 10 millimoles per liter per day. Is the total body weight meters multiplied by serum sodium divided by 140 minus 1. Here we have to also calculate the insensible water losses that is estimated to be about 10 ml per kilogram per day, and renal or extra-renal, let's say, gastrointestinal tract water losses, and also to calculate it in febrile patients about 3.5 ml per kilogram per day per 1 degree of Celsius if there is an increase in the temperature. Now, the preferred route for management of hypernatremia, as we all know, is to administer fluid by mouth or by a feeding tube. If neither is feasible of these two processes, fluids should be given intravenously. Hypotonic fluids, of course, are appropriate, like dextrose 5%, dextrose water 5%, or the half isotonic saline, 0.45. But can we use isotonic saline? Only in cases that there is a frank circulatory compromise, there is a hemodynamic instability. In these patients, we have first to restore the effective arterial wall. And after selecting the appropriate infusate, either will be dextrose water or half isotonic, or even other infusates can be used, the rate of infusion should be determined. Please upload question number 3. We have a 72-year-old patient who lives alone that is from the lab and clinical examination that it is confirmed that there is a diagnosis of euvolemic hypernatremia. There is no hypovolemia here. Which of the following is the most appropriate treatment? 5% dextrose water, loop diuretics, normal saline, sodium bicarbonate, or hypertonic sodium chloride of 3%. As we said before, in hypovolemic hypernatremia, we might use normal saline, but in cases of euvolemic hypernatremia, we have to give hypotonic fluid, and because of that, we should place the patient on 5% dextrose water. But how are we going to calculate the effect of 1 liter of any infusate we choose to use on serum sodium? How can we be sure that the reduction in sodium concentration would be less than 8-10 millimoles per liter per day? For this reason, this formula has been developed by Androgian Professor Nick Matias. Professor Nick Matias is a Greek professor at Harvard University, and this formula actually can calculate the change in serum sodium according to any infusate, to 1 liter of any infusate. It says that the change in serum sodium is the infusate of sodium minus serum sodium divided by total body water class 1, and also the second type of this equation can also calculate the change in serum when we also add to our infusate potassium chloride, because a lot of times these patients also have, as our patient, low potassium level. And as you see here, for 5% dextrose in water, of course, we know that there is no sodium there, so it is 0 millimoles per liter. For 1/4 of isotonic, for 1/2 of isotonic, for Rieger's lactate, you see that the sodium concentration of English lactate is 130, and for, of course, normal saline, we already know that is 154. The total body weight is calculated by this formula, 0.5 or 0.45 for women, multiplied by body weight. So, recapitulating about the management, the assessment of the extracellular body status is very important because patients with the decreased volume, that is by far the most common type of hypernatremia, should first be given isotonic saline in order to restore the effective arterial volume. The formula by Andrew and Matthias actually does not predict the duration of sodium in patients with hypernatremia and severe extracellular volume depletion, also when there is a marked reduction of renal support. However, this formula takes into account, as we said before, the amount of potassium added to the infiltrate is osmotically active sodium. For example, to see how important is the potassium chloride we might add to our infusate, 1 liter of isotonic normal saline plus 2 flacons of potassium chloride results in a hypertonic solution because you see sodium is 154, 2 flacons of potassium is about 27 because each flacon contains 13.5. So that results in 181 millimoles per liter of sodium plus potassium, and that results in an osmolality of 362. It's an hypertonic solution. So, with regard to the treatment of hypernatremia according to body status, in cases with a hypovolemic hypernatremia and the extracellular, the effective arterial volume and extracellular volume is low, we have first to give normal saline solution, and after we restore the effective arterial volume, increasing the blood pressure, see that there is urine output, enough urine output, then we might go and correct the hypertonicity with hypotonic solutions, dextrose water, or 1/2 or 1/4. In euvolemic hypernatremia, we should give free water replacement or intravenous glucose solution if it is possible. If it's impossible to give water by mouth, and in hypervolemic hypernatremia, that is not so common, free water replacement plus furosemide might also be used. Therapy should be guided by frequent monitoring of the plasma sodium and not by formulas alone. We have to send every 2 hours plasma sodium concentration in order to see if we have to get proper adjustment of the treatment schedule in order for the safe and effective management of patients with hypernatremia. The last question, we have a patient with a sodium level 168. It has been developed during the last 5 days, so it's a chronic hypernatremia, and we are ordered to start the patient on 0.45 sodium chloride, meaning half isotonic. What is the most important intervention? As we said, the most important intervention should be the not rapid correction of hypertonicity. So it is to insert the central catheter to give the solution rapidly, to reconfirm the order because hypotonic sodium chloride is contraindicated with hypernatremia, to give slowly and watch for signs and symptoms of cerebral edema, or to place a bladder catheter to monitor the output. I will go through this question very quickly because the time is not with us. So the right answer, of course, is to give slowly and watch for signs and symptoms of cerebral edema. And 2 more slides about 2 or 3 slides about our patient. Our patient was a hypovolemic hypernatremia, was associated with increasing session water losses, most probably because of a coexistent infection. You remember the leukocytosis, increasing FP. The total body weight was calculated to be 35 liters. The water deficit was 5 liters according to the formula shown before. And as we said, since our patient was accompanied by volume depletion, first 1 liter of normal saline solution was initially administered over 4 hours in order to restore, as we said, effective arterial volume, resulting in blood pressure elevation from 85 to 105/75, and there was enough urine output of 200 milliliters. Then we calculated for reduction, as we said, less than 10 millimoles in 24 hours. We had the plasma sodium, as you remember, 160. the total body weight was 35 liters. The renal water losses were calculated to be 500 to 600 ml. The insensible water losses to be 10 ml per kilogram per day so it was 70 kilograms, 700 ml. And because he was also febrile, there were also 270 ml added to this total. This total was 1.5 liters plus the 5 liters of water deficit. And I have created here according to the infusate that would be chosen, what would be the liters per day, first for dextrose in water, 5%. You can see it was 3.75 liters of dextrose in water, 5% in water, over 24 hours, that means 156. If it was chosen to give also 4 flacons of potassium chloride, then it was going to 4.91. If it was for half isotonic, it was 5.85 liters. And I have put here, just for your information, if we have chosen to restore the hypertonicity, to actually to reduce the sodium by 10 millimoles per liter per day, and we had to choose normal saline, we should have given to the patient 60 liters over 24 hours, meaning 2,500 ml per hour. As you can understand, this is unacceptable when the patient would have pulmonary edema. So concluding, hypernatremia results from a deficit of water in relation to the body sodium content, which can result either from net water loss or for hypertonic sodium gain. Barely. History, physical examination and labs are guaranteed to uncover the underlying cause. The type and the rate of infusion is of great importance, since overtreatment has been associated with increased morbidity and mortality. And the last but not least, treatment should be guided by frequent monitoring of the plasma sodium, and appropriate adjustment of the treatment should be mandatory to improve the outcome, especially in elderly and critically ill patients. I try to do my best to be as fast as possible, as quick as possible. Thank you for your attention.
Thank you, Professor Margaritsis, and this concludes our last session. Thank you very much.